Welcome to this explainer. Have you ever looked at a plant and wondered, how it actually survives? Or more importantly, how it keeps us alive? Today we're tracing the origins of your food all the way back to the sunlight captured by a single, perfectly engineered leaf. We'll be using this interactive whiteboard to break down the exact mechanics, the invisible structures, and those crucial formulas you absolutely need to know. Let's dive in.
Here's our roadmap for today. We're covering the magic of photosynthesis, anatomy of the leaf, the chemical journey, limits and variables, and finally, acing your exam.
Section 1. The magic of photosynthesis. It's honestly wild to think about, but literally every single calorie of energy you consume originated as a photon of sunlight hitting a green plant. Seriously, because this process is the absolute foundation of our entire ecology, and let's be real, a highly testable topic for your exams, we're going to visually dissect exactly how it works.
Section 2. Anatomy of the leaf.
Okay, notice the dimension lines drawn right across this dicotyledonous leaf. See this broad, thin, flat area? That is the lamina. It acts as the primary capture surface. Think of it exactly like a man-made solar panel. The wider the surface area, the more photons it can physically catch. Now follow these yellow directional arrows on screen. Watch how the sunlight actually travels. It first hits that broad lamina, then it penetrates the clear top boundary you see right there, the cuticle. This layer is an absolutely brilliant biological adaptation. It's completely transparent to let light in, but physically sealed to stop water from escaping. You can see the light vectors traveling directly down into the internal cells.
Look at this magnified insert bubble right here. We're zooming in to find these vertically aligned palisade mesophyll cells. Notice how densely packed they are with chloroplasts? Those green dots are the absolute powerhouses of the plant. They sit right at the very top of the cell layer to intercept the maximum amount of light coming through that transparent cuticle.
Section 3. The chemical journey.
Let's map the variables. We have a super clear input and output system here on our flowchart. Our balanced word equation starts with the reactants on the left side, carbon dioxide plus water. Now add in our crucial catalyst. That's the light energy captured by the chlorophyll. And following the arrow, this yields our products: glucose for stored energy and oxygen, which gets released right back into the air.
All right, let's stamp those exact chemical symbols onto the components, because you definitely need to memorize these. On the left, we have 6 molecules of CO2 and 6 molecules of H2O. Under the influence of light energy, watch them transform seamlessly on the right into one single glucose molecule, that's C6H12O6, and 6 oxygen molecules. Notice how perfectly the scale balances here? Every single atom is strictly accounted for.
Now, look at this split screen layout. This whole process builds into a 2-stage assembly line. First, on the left, we have the light-dependent reactions happening right in the thylakoid membranes. Here, light energy physically splits water molecules, creating these little chemical energy packets called ATP and NADPH. Then, follow the flow to step 2 on the right, the light-independent reactions, also known as the Calvin cycle, taking place in the stroma. The cool thing here? Direct light isn't needed. The plant just uses those energy packets we just made to power the conversion of carbon dioxide into our final glucose molecule.
Section 4. Limits and variables.
The rate of photosynthesis isn't just some static, constant number. As we can see from the dynamic labels popping up around our plant, it fluctuates. Light intensity, carbon dioxide concentration, temperature, water availability, and chlorophyll concentration all act like physical dials. If you turn any one of these inputs up or down, our entire chemical assembly line changes speed.
Let's track this graphically. Look at bar 1 and bar 2 on this chart. As the light intensity increases, the rate of photosynthesis shoots right up. But then, look over at bar 3. Notice that red boundary line where the chart just goes completely flat? Yeah, even plants have their limits. Increasing light boosts the rate only up to a distinct boundary. After that point, no matter how much more light you blast it with, the rate plateaus, usually because another factor, like temperature or CO2, is now holding the whole system back.
To prove this experimentally, scientists follow a very specific chronological workflow. Step 1. Destarch the plant in complete darkness. This is a vital control step to ensure any new starch we measure is purely from our experiment. Step 2. Expose the plant to specific controlled variables, like a test light source. And step 3, measure the active rate. By literally counting the oxygen bubbles releasing from a piece of pondweed and following their path upward, we get a real-time, physical metric of exactly how fast photosynthesis is happening.
Section 5. Acing your exam. Okay, let's talk strategy. You really want to avoid these common traps that examiners just love to test. First off, never mix up the physical locations. Light-dependent is in the thylakoids, light-independent is in the stroma. Second, remember that boundary line we just looked at? Infinite light does absolutely not equal infinite photosynthesis. Something else will eventually limit the process. And finally, remember that stage 2, the light-independent reactions, does not require direct light to operate. That's a huge one.
Your ultimate preparation tactic. Drill these variables. Literally write out carbon dioxide plus water yields glucose and oxygen. Then map it directly to the symbols. 6CO2 plus 6H2O yields C6H12O6 plus 6O2. Honestly, just practice mapping these until writing both equations becomes absolute second nature. You've got this.
I'm going to leave you with this final thought. We've traced the flow from a single photon hitting a leaf all the way down to the splitting of water in the thylakoid and the production of sugar in the stroma. So, based on the chemical limits we just mapped out, how would you act as the ultimate biological engineer? Seriously, how would you design the perfect, tightly controlled environment to push a plant's photosynthesis to its absolute theoretical maximum? Think about that, keep practicing those equations, and I'll see you in the next explainer.